Hi class, welcome to Unit 5, Bacterial Growth. And in this unit, we are going to revisit some of the concepts from lecture regarding microbial growth, and then we are going to focus on the measurement of bacterial growth as done by spectrophotometry. So just a reminder that when we talk about microbial growth, we are not looking at increase in size, we are looking at increase in numbers. And bacterial cells, as prokaryotes, they divide by a process called binary fission. So this is different from mitosis, when you have the nucleus and first the nucleus divides and then the rest of the cell. No, this is a much simpler separation or division in the sense that there will be first the DNA will be replicated, so you have two copies of the DNA, and then there will be a formation of something called the septum. This is a division appearing in the middle of the cells, and then the two cells separate. So it's a very simple process. It also, depending on how quickly it happens, then it can, it's dividing one in two. It can produce an incredibly large growth in numbers. So for example, E. coli, when it is at its most favorable environment, it can divide in as short as 20-something minutes. So you can imagine that each cell divides to 2 in 20 minutes. That means that we are going to have a really exponential and fast growth. Let's look at the bacterial growth curve. And this is something that you want to revisit in detail and what happens in each phase. So what this bacterial growth curve shows is, let's say that you start a bacterial culture, you place some cells, you inoculate cells in a medium, and then you let it grow. You are not adding new medium, you are not changing the cells to a new medium, just you follow what happens with the number of cells during that time. And as you can see from the y-axis, this is actually looking at the living bacterial cells. So there are 4 phases, and again, this is if we don't fresh up the medium or if we don't take away cells. So this is a culture that has started and you're just following it as it goes. So in such culture, you're going to observe 4 distinct phases: lag, log, or exponential, stationary, or death and decline. So let's go quickly through each of these phases. So the lag phase, as you can see, there is no real increase in the number of living bacterial cells. And we can say that the lag phase is when the bacteria are preparing to grow. And depending on the condition, this can be a very short phase or it can be longer. So for example, if you are placing a cell from a certain medium into the same medium and the cells are growing happily before that, the lag phase is going to be extremely short because nothing has changed. On the other hand, if you place cells that were growing, let's say in a medium with glucose into a medium that has no glucose but instead has lactose, it needs some preparation to be able to metabolize lactose. And an extreme case would be when you are starting from endospores. You may recall that endospores were these resting survival structures of bacteria. Well, once they germinate, so this is when they are placed in conditions that are favorable again, this is going to take some time for them to get going because basically they are dormant cells. Okay, so lag phase is, when we look at the graph, it's going to be basically a horizontal line. There is no increase in number of living cells. The next phase is the log or exponential phase. And this is kind of what we were referring before of how quickly cells can divide if they are dividing, especially in a very short time. So cells here are happy. They are dividing quickly. So you're going to observe a logarithmic increase, an exponential increase in the number of cells. But again, we haven't done anything to that culture, so whatever nutrients there were in that culture medium, they start dwindling. And also the presence of so many cells may cause, toxic materials may accumulate, and then some cells may start to die. So what you observe in the graph is that there is a plateau. So there is no net increase in number of living cells, because some cells are dying, but some cells are still dividing. And compare lag with stationary phase. So they look the same, but there are very different things happening in both. So in lag phase, they are basically, not doing anything. The cells are preparing, they are alive, but they are not growing. In the stationary phase, that flat line reflects that some cells are living, some cells are dying. So basically, the number of cells dying and dividing is roughly the same. But again, we haven't added fresh mediums, so there is no more food. There is just too many dead cells, toxic products. So then we come to the last phase, which is the death of decline phase. So now the cells start dying at an accelerated rate. So it's a little bit of the mirror image of the log phase. So it's also an exponential change in numbers, but this is in the other direction. And if a cell is able to make endospores, this would be the time when they would be making endospores. So this is a long slide, long text, so I'm not going to read it, but this is kind of how would you do a bacterial growth curve experiment in the lab. Basically, you start with a single colony of bacteria inoculated into a broth. You grow that broth a little bit, so you have some kind of numbers of cells, and then you inoculate it in a flask with a larger volume. So in this case, this is 0.3, so you're going to have it in a 250 ml of broth. Then you add 5 milliliters of this original broth, and you are going to start measuring optical density. So I'm going to, in the next slide, to talk about spectrophotometry, but optical density in this case is going to reflect the turbidity, the cloudiness of your broth. So clearly, if you have very few cells, then your broth is going to be more or less transparent. But as you have more and more cells, your broth is going to be cloudier. So by measuring the optical density at a certain time interval, in this case every 30 minutes, you'll be able to plot those numbers. And ideally, you would be following it in the exponential growth curve. This is a picture of a spectrophotometer. So spectrophotometry is based on a beam of light of a certain wavelength is passing through your sample. And then in this particular case, we are using 600 nanometers. So the clearer the solution, like the one you see on the top, then your optical density is going to be smaller, and then the higher the turbidity, then your optical density is going to be higher because it's cloudier. So we are talking here about optical density, which is the same as absorbance. You could measure it as transmittance, which would be, how clear the liquid is. So it's two sides of the same thing. Here's an example. This actually came from a real lab. Cells, I believe it was E. coli probably. So E. coli were placed in broth at different temperatures. So you can see that the blue is 15 Celsius. This is pretty coolish. Then you have 25, which is room temp, 35, 45, and even 55. And the growth curve was done. So to the left, you can see the absorbance values, and then they were plotted over time. and which is the best temperature. So you would be looking at the graph, and you can see that the black graph is going the highest. So 55 Celsius would be the most favorable, the optimal temperature for this microbe. Let's talk about another aspect of how bacteria grow. Bacteria can have very different oxygen requirements. And we have been talking about aerobes and anaerobes. So if we look at this diagram here to the very left, so if a bacterium is what we call an obligate aerobe, that means that that bacterium can survive without oxygen. So if you inoculate it in, let's say, an agar deep or even in a broth, the bacteria is going to grow close to the surface so it can access oxygen. Let's move to the cube number 4, so fourth from the left, which says obligate anaerobe. Well, that would be the opposite. So this is a bacterium that not only cannot use oxygen, but also would die in the presence of oxygen. And that would be the case, for example, of Clostridia. You may recall that Clostridia are obligate anaerobes. They do not have catalase. So they tend to grow in the very bottom of the tube to be as far away as they can from oxygen. So let's look now at tube number 2 and number 3. And let's look at the pattern. So you can see that they are growing everywhere. But the difference is that the facultative aerobes, actually, sometimes you can find them as facultative anaerobes. They prefer oxygen. They can grow without it, but they prefer to use oxygen. So these facultative aerobes are going to be closer to the top of the tube, while the aerotolerant ones, they don't use oxygen, but they don't mind. They don't care. So that's why there is no difference in the distribution of the bacteria. And the very last one has a very unique pattern, as you can see. It's called the microaerophile. And these microaerophiles, they use oxygen, but use it as a lower concentration than the oxygen present in the atmosphere. So they are going to grow at a little bit further away from the oxygen, but not everywhere, because they still require oxygen. So how does this look in real life? Well, in real life, for this kind of experiment, you would use a special broth called the thioglycollate broth. And thioglycollate broth is a broth that contains a chemical that can capture oxygen. So in the very bottom of the tube, or inside the broth, it is quite anaerobic, the environment. And it has a little bit of agar in it. It's not a lot. It's not a solid medium, but it provides a little bit more thickness to the gel. So the tubes are labeled A, B, C, D, and E. And I would say that the easiest to identify is number D. So that really corresponds to the microaerophile that we saw in the diagram. And this is a Neisseria. You may recall Neisseria, it's a gram-negative coccus. So Neisseria sicca is not any of the nasty Neisseria, but this is microaerophile, so you can see that it grows just below the surface of the media. Going to E now, in the tube E, we have Pseudomonas aeruginosa, which is an obligate aerobe. So you can see how there's nothing, there's no growth in the bottom of the tube, and most of the growth is concentrated in the very top because it requires oxygen to survive. Conversely, tube B would be the obligate anaerobe. So this is a Clostridium butyricium. And you can see there is no growth in the very top, and most of the growth is from kind of the 2/3 and down. So the ones that remain are number A and number C, and they are both called facultative anaerobes in this diagram, although they look slightly different, but they both grow everywhere. And I'm going to go back here. This slide say facultative aerobes. It's actually facultative anaerobes. So remember, facultative anaerobes are going to grow everywhere, but they tend to be more prevalent closer to the oxygen. So, so it is a little bit harder to see in this picture here, but there is a little bit more growth, especially when you look at tube A. There's a little bit more growth closer to the top than to the rest. And in the case of C, well, the growth pattern is a little bit funky, but again, this is not something that you're going to get in an exam. A picture like that, you will get a diagram, but it is growing everywhere, but a little bit more towards the top. And there is no error tolerant in this particular picture. Okay, so now I'm going to change completely the topic, and I'm going to talk about plaque assays. So plaque assays, because there is a video about it in the unit, and it talks about growth, but in this case, it's growth of viruses. So bacteriophages are a kind of viruses that infect bacteria. And if you want to quantify viruses, well, one of the big issues is that viruses don't grow by themselves. You have to infect cells in order to measure, or in order for them to divide. But that ability or that characteristics can be used to detect them, in the case particularly of bacteriophages. So let's say that you have this petri dish, like you see in the picture, which has a bacterial lawn. So all that cloudy background on the plate is just bacteria that have been spread and grew on the plate. And then you add viruses to these phages. And if a virus infects a bacterial cell and it kills it, it replicates and then it bursts out of the cell, those cells die. So you're going to see them as this clear little spot on the slide. So this is what we call plaques. This would be this little clear spot on the plate. And a little bit similarly, like we did with serial dilution when we were counting colonies last week, that you were counting colony forming units. In this case, you will be counting plaque forming units. So you'll be doing the same procedure, which would be a serial dilution of your viral sample, because you don't know how many viral particles there are, and then you plate them on different plates, and you look for a plate that has plaques that can be counted. So you'll be looking at a plate with, let's say, between 25 and 150 plaques in them, so those are easier to count, and then you will be multiplying with whatever dilution factor you use. So it's the same procedure that you'd be using for CFUs, but in this case you are counting plaque forming units, not colony forming units. And as you can see, this is a pretty brief material for Unit 5. Thank you.